Has been a while, and I promise you, I'm still alive, I'm still here. I've just been really, really busy, and also I've covered the entire biology syllabus for you, so I really try to avoid just doing videos for the sake of doing them. I want to make sure that whatever video I do is a video that can help or be of help to you in some way. So that's why you haven't heard a lot from me. For those who keep sending messages and emails about the syllabus, I want you to know that yes, indeed, all the content on the channel can be used with the new syllabus and will likely be used with the syllabus that comes after that. And that is because the A-level biology syllabus, even though it changes, doesn't exclude the core information. And most of the time, the syllabus changes are more like things are being removed from the syllabus, so there are things you don't need to study as opposed to things being added. So you can definitely use all the videos on the channel for this. So I want to take a new approach with this video. I would like you, if you haven't practiced this paper and you're writing the October-November 2022 exam for paper 1, I'd like you to first of all just give the paper a shot, right? Like, so take the paper, answer all the questions from 1 to 20, because this video will only cover numbers 1 to 20. Once you've answered all of the questions, then you can watch the video and then watch the explanations just to see if your thinking around the questions was correct, what you did wrong and what you did correctly, things like that. I also want you to know that paper 1 is often overlooked by a lot of students because students assume that if it's multiple choice, it is easier. It is not easier. It is one of the papers where, in my experience when I was working as a teacher, students failed a lot because they underestimated it and they assumed that the questions were very direct. Paper 1 requires you to think just as deeply as paper 2 would, and I would offer that paper 2 is in fact easier than paper 1 because in paper 1 you can either get options that look really similar to each other or nothing seems to be answering the question if you don't have the in-depth understanding of the biology. So we will go over numbers 1 to 20 of this paper in this video. And that's because I don't want the video to be too long, and we will take it from there. So let's give it a shot. All right. Okay, so I'm hoping that by now you have answered all the questions from 1 to 20. Just take 30 minutes to do that before you watch the video, and now you are here to watch the video. Okay, so starting with question 1 says a student was asked to use the scale bar to show the scale bar shown to calculate the magnification of a cell on a photomicrograph, and this is the scale bar. As you can see, the scale bar has like 2 vertical lines at the ends, and that's basically where the bar starts and ends, right? And then it has a number under it. Then the question asks, which method could the student use to calculate the magnification of the cell? Now, with questions like this, I usually tell students, just think back to the technique that you remember or the technique that you know. Write everything you know about this question. So the first thing we know for sure is that magnification is equal to image size over actual size. Okay. And just to warn you ahead of time, my pen has been acting a little bit funny, but I hope that it takes us through this video. I need to possibly get a new pen. Okay, so magnification is equal to image size over actual size. We know that. Now, when you're given a scale bar next to a picture, so let's assume this is a picture or a cell, right? You're given the cell, and then you have a scale bar next to it, and under the scale bar or on top of the scale bar, doesn't matter, there is a number. So let's say like that. How do you determine the image size and the actual size from the scale bar? First things first is for you to know that the actual size is the number under the scale bar. Okay, so the actual size in this case is 2 micrometers. But then what is the image size? I've seen students take their ruler and try to measure the length of the cell. If you have a scale bar, you don't need to measure the length of the cell. What you need to do is you need to take your ruler and measure one end from one end of the scale bar to the other end in millimeters. Okay, so you measure that using the millimeter part of your ruler. You convert millimeters to micrometers, and that means you multiply it by 1000. Okay, so that would give you the value in micrometers. So let's say, for example, this was 10 millimeters. In micrometers, it would be 10,000 micrometers because you multiply that 10. All right, and then that will give you what the magnification is. So in this case, they're not asking you for specific numbers. So again, don't get carried away and start to calculate the numbers. They're asking you what method you would use. So option A says divide the diameter of the cell by the length of the scale bar with both measured in the same units of length. That's wrong because they didn't tell you anything in the question about the diameter of the cell. Okay, also when you're given a scale bar, you don't need to measure the cell. Measure the diameter of the cell in millimeters, multiply by 2000 and divide by the length of the scale bar measured in millimeters. This method is just absolutely wrong. Firstly, because the number 2000 is a very ambiguous figure. There's no indication of where that has come from. So that's not your answer. C says measure the length of the scale bar in millimeters, convert to micrometers and divide by 2. That is exactly what I've explained to you here, okay? So this is our likely option. Measure the length of the scale bar in millimeters, convert to micrometers, and multiply by 2. That would be wrong for the formula of magnification. So remember, magnification is image size over, this is like a slant here, actual size. So it's a division, it's not a multiplication. So that tells you that the answer for number 1 is C. All right. Let's look at number 2. Which statements about light microscopy are always correct? All right. So first statement says the greater the resolution of a light microscope, the greater the detail that can be seen. That's true for any microscope at all, that if you have a microscope with a high resolution, it basically means that you can see a lot more detail, right? And remember that resolution is being able to tell detail, being able to see more detail, or being able to tell features apart. So yes, this would always be correct, that when resolution is high, more detail can be seen. The second statement says the greater the magnification of a light microscope, the greater the detail that can be seen. Okay, so this one is wrong, and I'll tell you why. When you're dealing with a light microscope, you sacrifice resolution for magnification. Now, what does that mean? When you increase your magnification, okay, you start to decrease your resolution. What this simply means is, say I have a light microscope and I put it at 1000 times. I won't see, I might not see a lot of big detail, like in terms of like the size, it might not be big. But I'm likely to see more features of the cell. So I might be able to see, like, some of the ribosomes moving around and so on. The moment I take this magnification and I go to, let's say, 100,000 times, what I will notice under the microscope is that the resolution is reduced, which means that the image becomes a little bit blurry. So I want you to try it when you're in school, to just go and have a look and start with like a small magnification. Look through the microscope, have a look at what you see and how much detail you can see. And as you increase the magnification, look at what happens to the resolution. You'll realize that the resolution becomes poor. And resolution is basically the amount of detail you can see. So in this case, number 2 is wrong. Increasing the magnification of a light microscope up to its limit of resolution allows more detail to be seen. That is correct. Which means what the limit of its resolution means is that it's maximum resolution. So if you increase the magnification up to the point where the resolution is maximum, you will see more detail. But once you exceed that limit of resolution, then you start to see less detail. So there's a point, I think, in the light microscopes that you have at school. There's like a 4 times objective lens, there's a 10 times objective lens, and there is a 100 times objective lens. All right, so there's a chance that at 10 times objective lens plus the 10 times of the what's it called now, plus the 10 times of the eyepiece, you're going to see, quite a bit, right? Because you make it 100 times bigger. In this case, when you add that 10 and it's 1000 times, the resolution might be poorer. Now, number 4, the shorter the wavelength of light used in the light microscope, the greater detail that can be seen. Yes, that's true. The shorter wavelengths actually produce more detail for the light. So we see now that 1, 3, and 4 are correct. So our answer here is B. Let's look at 3. What is the length of a typical prokaryote, such as the bacterium Escherichia coli? This just reminded me of when I was in undergrad and I had to learn the names of these microorganisms because I did a BSc honors in molecular biology and biotechnology, and I worked a lot with bacterium. We were required to know their names and their features, as well as those that were disease causing and the impacts that they could have if they infected a person. So really interesting stuff. But that's not necessary to answer this question. I was just giving you some extra detail. So the length of a prokaryote typically is between 1 micrometer and 2 micrometers. Okay. So in this case, because we have 1.5 as the option, so we're just going to go with 1.5 micrometers, right, being the answer. But now you have 1.5 times 10 to the power of 1. That means 1.5 times 10, and it's also in nanometers. So the fact that it's in nanometers already marks it wrong. So I would go ahead and just cancel out the ones that are not micrometers. Now this one is in micrometer, but 1.5, and this is something, please pay attention to this, that students tend to miss out on, or they make a mistake because they're in a hurry. 1.5 times 10 to the power of 1 is 1.5 times 10. And that gives you 15, which is the wrong answer. The right answer is 1.5 micrometers. Any number that is raised to 0 in mathematics is equal to 1. So this answer here is 1.5 times 1 micrometers, and that's 1.5. So your answer is C. I hope that was clear. Number 4, which cell structures are present in Plasmodium? I hope everyone can recall that Plasmodium is the organism that is responsible for malaria. And Plasmodium, again, another error a lot of students make is they assume that Plasmodium is a prokaryote, but it's not a prokaryote at all. It is actually a protoctist. Okay. And a protoctist is not a prokaryote. It has eukaryotic features, right? So Plasmodium is, well, you can just consider it to be a eukaryote. So in that case, when you're looking at the cell structures that would be present in Plasmodium, you have to think of it as looking for the cell structures that would be present in a eukaryote. So would a eukaryote have ribosomes? Yes. Would a eukaryote have mitochondria? Yes. Would a eukaryote have Golgi body? Yes. Now, I know these Venn diagrams for some students can be quite confusing because they're not really sure what it means. So the best way to explain it to you is that whatever option you see on beside the circle, that's the option it's pointing to. So if you were to choose B here, you're saying that only ribosomes would be in I'm just going to clean that out. Only ribosomes would be in Plasmodium, okay? If you choose D, you're saying only mitochondria. If you choose A is at the intersection of B, C, and this circle here, which is the Golgi body, okay? And we know that all three of them would be found in Plasmodium. So that means the correct answer is A. If you choose C, you're basically saying it's only the intersection of these two circles. So that's ribosome and mitochondria, and that would be wrong. All right, let's look at number 5. Which cell structures may contain nucleic acid? I really love these questions. For some reason, it seems as though CIE came back with a bang after the pandemic interrupted exams for a while. But what are the nucleic acids? And these are the things that make students fail because they're not really thinking deeply about what they should be answering. So some students just jump right into it and they make a mistake. So what are the nucleic acids? Your nucleic acids are DNA and RNA, at least for your syllabus and for biology, as much as I know it. And by the way, I'm not do not consider me to be the authority on biology. There are people way more qualified than I am, because after my undergrad, I did a PhD in engineering. So Much of my knowledge is based on undergraduate biology still. All right, so DNA and RNA are the nucleic acids, and how do you know that? The full meaning of DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. All right, so where would you find DNA? I think that's where you need to start with. Like, in which of these organisms would I find, which organelles or cell structures rather, would I find DNA? So you wouldn't find DNA in the Golgi body, you wouldn't find it in the ribosome, you wouldn't find it in the cytoplasm, but you would find DNA in the chloroplast. And why is that? That's because of a theory called the endosymbiont theory. I really want students to have this theory at the back of their minds when they answer questions like this. The endosymbiont theory basically states that the chloroplast and the mitochondria used to exist as independent organelles, or independent organisms rather, before they were engulfed by a eukaryotic cell, leading to the establishment of a symbiotic relationship between the eukaryotic cell and these organelles. So chloroplast, if it was existing on its own, means that it had its own DNA, its own protein making machinery, and all of that. So yes, you would find DNA in here. Okay. Now, where would you find RNA? We've done the list for DNA, and we know that DNA is not anywhere else. DNA for eukaryotic cells, for the most part, is in the chloroplast, in the mitochondria, and in the nucleus. Nowhere else. For RNA, where would you find RNA? Now, you can find RNA in the cytoplasm. And why is that? Because when the RNA polymerase, and if all of this is sounding like jargon to you, please just watch the videos on chapter 6. When RNA polymerase goes into the nucleus to copy the DNA into RNA, that mRNA comes out into the cytoplasm to attach itself to a ribosome. So you can find RNA in the cytoplasm, and you can find it on the ribosome. Now, would you find RNA in the Golgi body? The answer is no. The Golgi body only functions to fold proteins after they have been made. Okay, so it modifies proteins after they've been made and released from the ribosome. So in that case, if we know that 1, 2, and 3 are correct, then our answer is going to be A. All right. Now let's look at question 6. Which row shows correct features of ATP? ATP is adenosine triphosphate. Please do not confuse adenosine with adenine. Adenine is what you find in DNA and RNA. Adenosine is a different compound altogether. This question requires you to know the structure of ATP because it says you have to know the carbohydrate. Looking at the answers that you have here, there's a bit of confusion, and I would understand if students were to accidentally pick the wrong answer here. And the reason for that is that deoxyribose and ribose are 5-carbon sugars. So they are both pentoses, okay? The reason why you can't pick one or the other is probably here. So obviously if you say, well, it's not because they're deoxyribose, well, obviously. But yeah, mitochondria and chloroplast is where you would make ATP. Mitochondria only, that's not true. Chloroplast only, that's also not true. The sugar that's in ATP is not deoxyribose. Deoxyribose is the sugar in DNA. So that's what makes this one wrong. Okay, that leaves you with just this option, pentose. So I don't know if you see what I did there. I used an elimination technique to remove the options that were not possible simply based on what I know. So again, if I want to go over it with you, I'm just going to erase these. So if you have a question like this where it's confusing because the options are so similar, deoxyribose is a pentose, ribose is a pentose. A lot of people would say, okay, if all of these are pentoses, then the obvious answer is hexose and chloroplast only. But you know that ATP is also made in the mitochondria. And if you didn't know that ATP is made in the chloroplast, now I'm telling you. When we talk about the chloroplast, we tend to talk about it in terms of sugar production during photosynthesis, so it makes glucose. But that's not the only function of the chloroplast. It actually makes ATP as well. So you know that, okay, this can't be the answer because chloroplast only is not an option. Mitochondria only is also not an option because, like I said, it's in both. Deoxyribose is the sugar in DNA. It's not the sugar in ATP, so that can't be the answer, and that leaves you with B. All right. Let's look at 7. Which test will identify biological molecules that contain monomers with a carboxyl group? Again, I think CIA just came back with a bang, and I'm loving these questions because they really make you think as you prepare for the exams. So first things first that you want to think about, if I was approaching this question, is I would look at the options I've been given. So I know that ethanol is for testing lipids. Okay. I know that Benedict is for testing carbohydrates or reducing sugars. I'm just going to put there as carbo. And Biuret is for testing proteins. Okay. So now that I know all of these three, which of these have a carboxyl group in their structure? Definitely not carbohydrates. Okay. Carbohydrates have CHO as their structure, CH2ON as their structure, and there is no part of that that forms a carboxyl group. For those who are wondering what the carboxyl group is, it is COOH. Proteins, on the other hand, are made up of amino acids, and it's asked you which one will contain monomers with a carboxyl group, or which one will identify biological molecules with monomers with a carboxyl group. This is a monomer of carbohydrates, and it doesn't have a carboxyl group. Proteins, on the other hand, because they're made from amino acids, they have a different structure, and I'm starting the structure wrongly. Clearly, I'm a little bit rusty here. So, for those who remember how I explained this, you have an amine group at the one end, who I like to call Amy. Okay. So this is Amy on one end. This is a side group. This is a hydrogen or a pastor or imam or whoever you want to refer to that as. And this here is a carboxyl group. And I'll tell you why I'm saying all these weird names I'm saying now. Okay. So the way I help my students remember the structure of an amino acid is I tell them this is Carbo. Okay, Carbo is a nice guy. He's married to Amy. Amy is a nice lady. Because diamond rings are made from carbon, so they have a carbon between them. This is the pastor who's joined them together, or the imam, or the ceremonial director, whichever one aligns with your beliefs around marriage or wedding ceremonies. And this is unfortunately a side chick in their marriage. Okay, so that's the side group. So this is how I help my students remember the structure of an amino acid. So here already you can see that that has a carboxyl group. So C is a possible option. Lipids also have a carboxyl group, and the carboxyl groups are usually at the end of the chains. So they're usually long chains, and at the end they have carboxyl groups. So you know that this is also an option. So you need to find where these two are intersecting with each other, and that is with B. Okay, and that is your answer. All right, let's look at number 8. Which polysaccharide is correctly described? Okay, so A, the polysaccharide is amylopectin. Amylopectin, I mean, already I can see that this is a mistake. Amylopectin is not made with beta glucose. Beta glucose is a monomer of cellulose. So already you can just mark that one wrong. Amylose is made from alpha glucose, yes, and it is used for storage. It's used as a storage molecule in plants. That's a possibility. Cellulose made from alpha glucose, that's wrong. Okay. Glycogen made from beta glucose, that's also wrong, because glycogen is actually made from amylose and amylopectin. And so in this case, that would be B, because it's a storage molecule. So Something you should bear in mind, the difference between amylose, amylopectin, and cellulose is that amylose and amylopectin are storage molecules. So they're used to store sugars in the form of perhaps glycogen, because I think glycogen is made up of the combination of those two. Cellulose is a structural molecule made from beta glucose, which means cellulose is not used to store sugars in plants or animals, but rather it is used to provide structure to plants. Okay. But the reason why it's wrong here is because it says it's made from alpha glucose. So that's wrong. Let's look at number 9. Which formula represents a saturated fatty acid? This is really an interesting question. So a saturated fatty acid means that you would have all your carbon bonds occupied. Okay, all your carbon bonds will be occupied, and your carbon is able to form 4 bonds. Already I can see what the answer is, because I mean I'm looking at it and my chemistry is still alive. Definitely can't be this one. If you have 18 carbons, so let's just look at it. If you have 18 carbons, okay, you will need more than double this in order to, you need at least double the number of carbons you have in order to fill up the whole thing and make it saturated. A saturated fatty acid is basically a fatty acid that doesn't have double bonds within the chains. So if you have only 34 hydrogens, you already know that there's a high chance of you having double bonds. So that makes this answer wrong. And if you look at all of these, they have the exact same trend. So that makes the answer wrong. So your answer is possibly this. I've used a very quick technique here of estimating, right? I know for some students, they might want to spend time on this and draw out all the 36 carbons and count the hydrogens. A good way to look at it is to think of the carbon that starts at the one end as occupying 3 hydrogens, or rather having 4 bonds. But from then on, it doesn't only bind to hydrogen, it binds to another carbon. That carbon binds to 2 hydrogens, binds to another carbon. So it's a lot of work if you're trying to do the estimation, but that way. The best way is to look at the options that you have and just ask yourself, if I'm trying to not have any double bonds, then I need to have as many hydrogens or as much hydrogens as possible to fill up the carbons, right? And so if I have 18 carbons, there should be at least double of that, and that's what you have here. And in this case, you don't even have up to double of that, so that would be wrong. I hope that made sense. If it doesn't, ask me a question in the comments. I promise you I'll respond. Let's look at number 10. Which molecules always contain at least 4 bonds? So it has triglycerides, collagen, and hemoglobin. Collagen and hemoglobin usually have at least 4 bonds, and that's because of their structure. So hemoglobin, remember, is made with 4 polypeptide chains. So those polypeptide chains come together, so you can assume that there are at least 4 bonds in there. Collagen is actually made with into a triple helix, but the proteins also have like bonds that they form with each other. So it's also like a quaternary structure. Triglycerides, yeah, but the question, at least 4 double bonds. Oh, I'm sorry. I was thinking, why are they asking for 4 bonds? That makes no sense, because for collagen, it wouldn't make sense because it only has 3 chains. At least 4 double bonds. Well, with a triglyceride, it's hard to tell. A triglyceride doesn't have to have any double bonds at all. It can be saturated or unsaturated. So to say always contain, then this option would be wrong because of the triglyceride. This option is also wrong because of the triglyceride. This option is also wrong because of the triglyceride. Hemoglobin and collagen are likely to contain at least 4 double bonds. And so that leaves you your answer as C. And if you want to remember how this works, just go back to biological molecules where I explain the structures of these compounds. Let's look at 11. Which row about the structure of proteins is correct? So the primary structure here for A, it says the number of amino acids present in a protein. That's not true. So the primary structure is not about the number of amino acids. The number of amino acids can be as long or as short as is required. It's the sequence, so the arrangement of the amino acids in the protein. Okay, so that makes this wrong. And for me, when I'm dealing with questions that have this much detail, if I find one wrong option in one of the choices, then I just mark that entire choice as wrong. I can always go back to it to make sure my answer is correct, but it's how I optimize for speed. Okay, so B, the order of amino acids present in a protein encoded by DNA. So, I mean, I would put a star here. I know it's not the right answer, but I'll put a star there. Secondary structure, the coiling of a chain of amino acids to form a beta pleated sheet. This again is not wrong, but it's also not fully correct, because a secondary structure is not only about forming a beta pleated sheet, you can also form an alpha helix. Okay, so this again, not so much. Tertiary structure, the shape formed by folding of a polypeptide and held together by hydrogen bonds. This again is not fully correct, because in the tertiary structure, it's not only the hydrogen bonds that matter, but you have the disulfide bonds, you have the hydrophobic interactions, and you have other bonds that interact within the protein. So based on that, I will go ahead and actually cancel B as an option, because even though some elements of it are correct, they are not fully correct. Let's look at C, the primary structure, the result of translation of an mRNA molecule by a ribosome into a chain of amino acids. You know what? That's a possibility. All right, so we can put a star there. Secondary structure occurs because of attraction between hydrogen and oxygen atoms in the side chains. That's not true. That's actually not true at all. The tertiary structure, the result of ionic and hydrogen bonds, disulfide bridges, and hydrophobic interactions between amino acids. This is correct. But you see now, this one is wrong. So I'm just going to mark that as not right. So that means D has to be my answer. So let's go and have a look at it. Primary structure, the sequence of amino acids in a protein coded by an mRNA molecule. That's correct. Again, watch chapter 6 to really understand this, because as you can see with the way CIE asks their questions, they pull from even later chapters into the earlier chapters. So be prepared. I feel like 2022 will follow the exact same format. The secondary structure formed by hydrogen bonding between amino acids forming the primary structure. Yes, that's correct. Okay, so it's hydrogen bonding, not necessarily bonds between hydrogen and oxygen atoms. Let's see, and it says here it says in the side chains, and it's not really about the side chains actually. Then the tertiary structure formed as a result of interactions of the side chains of amino acids in the primary structure. That is also correct. So a lot of the folding that happens because the side chains start to interact with each other, and that's what leads to the tertiary structure. Wow, look at that. We've spent half an hour here already. Let's see if we can get through the rest of this a bit quicker. Which statement about collagen is correct? So that's number 12. A collagen fiber is made of 3 parallel helices with hydrogen bonds holding them in place. The helices are not parallel. So when you say it's parallel, that means you're imagining the collagen fibers are just literally standing next to each other like that, and all 3 of this would make one collagen structure. But they interact, they actually form like they coil around each other like that. It says 3 parallel helices. So it's basically saying this is one. I mean, there's so many ways you can think of this, but that answer is wrong. Number B, or option B, it is an insoluble fibrous protein with a quaternary structure. That's absolutely correct. And remember that a quaternary structure is not a shape. A quaternary structure is what you have when you have 2 or more polypeptide chains making up the protein. Number C, or option C, 1/3 of the amino acids making up collagen are valine. That's not true. As a matter of fact, I think what you find in collagen is glycine, and that's because glycine is the smallest amino acid, And because it's small, it allows for the helices to form because it's at that point where it bends so that the fibers can come close to each other like that. Collagen fibers are formed from several collagen molecules held together by ionic bonds. That's just a made-up statement. So your answer here is B. All right, let's look at 13. Which properties of water are the result of hydrogen bonding between water molecules? So between water molecules, you have addition and you also have cohesion, right? So you have the forces of cohesion rather between water molecules. Between water molecules and the surface, you have the forces of adhesion. So in this case, let's see, solvent action. Not necessarily, because the solvent action, if you're talking about the forces of cohesion, which is what this is asking about, then it wouldn't really help with solvent action, because it means the water molecules are just sticking to each other and they're not trying to interact with other molecules. When water is acting as a solvent, it's because its molecules separate from each other sort of and interact with the molecules of whatever you're trying to dissolve in it. So it could be a sugar, it could be salts, it could be any kind of compound at all that is water soluble, and that is what would happen. So I wouldn't pick solvent action as the answer. Specific heat capacity, yes, because the water molecules stick to each other, they're able to hold on to heat a little bit longer. Latent heat of vaporization, I would also say that's true, because the water molecules are sticking to each other, then it takes a lot of energy for you to vaporize water. So in that case, 2 and 3 are the answers, and so the option is C. Okay. Okay, let's look at at number 14. Which descriptions about all enzymes are correct? So all enzymes. Number 1, catalyze the breakdown of large molecules into smaller molecules. That's not really what enzymes, I mean, like that's a very primary level definition of an enzyme, because you're like, yeah, it lowers the activation energy, which is in fact more accurate. But a lot of, at primary level or like at lower level biology, a lot of the understanding is that enzymes are these things that break down large molecules into smaller molecules. That's not necessarily the case. Enzymes more like they bind with a substrate and, you know, help the formation of products by reducing the amount of energy required. So I wouldn't pick that as an option that should go here. They only function inside of cells. That's also wrong, because you have enzymes in industrial processes. For example, even in the making of beer, in the making of yogurt and all of those things, you have enzymes that are not functioning inside cells. They form temporary bonds with the substrate. That's true, because they form the enzyme substrate complex. You should watch the videos on chapter 3 if you haven't. And they have a tertiary structure. Yes, enzymes mostly have more than one polypeptide chain. And also because they form an active site and a binding site and all of that. And that requires more than 1 or 2 chains. So that requires more than 1 chain rather, not more than 1 or 2. So in this case, our answer is D. All right. Let's look at 15. The cells in the roots of beetroot plants contain a red pigment. When pieces of root tissue are soaked in cold water, some of the red pigment leaks out of the cells into the water. Already you can tell this is like it sounds like an osmosis question, but I don't think that's what it is. An experiment was carried out to investigate the effect of temperature on the loss of red pigment from the root cells. It was found that the higher the temperature of the water, the higher the rate of loss of red pigment from the root cells. Which statements could explain this trend? Number 1, enzymes in the cells denature as temperature increases. Enzymes in the cell, so the pigment can no longer be used for reactions inside the cell and diffuses out. I wouldn't pick that as an answer. I wouldn't pick that as an answer, because there are a lot of assumptions in this. By saying enzymes in the cells denature, you don't know if the temperature was increased beyond the optimum of the cell's enzyme, so you can't really say. As the temperature increases, the tertiary structure of protein molecules in the cell surface membrane changes, increasing the permeability of the membrane. Now this is highly likely, right, that as you increase the temperature, if the pigment molecules are inside the cell, when the cell wall becomes loose, those pigment molecules will escape and leak out into the water. Phospholipid molecules gain kinetic energy as temperature increases. That's true. Increasing the fluidity of the phospholipid bilayer and allowing pigment molecules to diffuse out more easily. That's also true. So 2 and 3 are the ones I would take as correct, and that would be B for number 15. Okay, we are powering through. We have 5 questions including this one because I'm only going up to number 20. The diagram shows a partially plasmolized plant cell, and it says what is found at Z. And you can see the label. So, I mean, when you have a plasmolized cell, by the way, it means that you have placed your cell in a solution with lower water potential. Okay, so there's lower water potential on the outside. I'm just going to write it as H2O part. There's lower water potential on the outside, which means the water concentration in solution Y is lower than what's inside the cell. And as a result of that, water will move out of the cell into solution Y, which then means that solution Y will likely increase in volume and might start to fill the spaces outside of solution X. So what is found at Z in this case would be solution Y. And this is something interesting you can investigate in the lab if you want to, or if your school allows you to, because it's really fun to do these osmosis experiments. Okay, we have one of the very detailed questions here. And this, by the way, students typically, in my experience, when I was teaching, when they see questions like this, they immediately lose heart and think that the question will be difficult. So if you're one of those students, try to stay with me. I will not spend more than 5 minutes on this question, or I will try not to. So let's see how well I do. A single-celled organism lives in fresh water. Water that enters the cytoplasm of the cell by osmosis is collected into a structure called the contractile vacuole. To remove the water, the contractile vacuole fuses with the cell surface membrane. A student counted the number of times the vacuole filled and emptied when the cell was placed in solutions with different water potentials. The results are shown in the table. So just to explain that in plain language, this is a single cell organism, all right? It's living in a water environment, okay? So there's lots of water around it, and it's fresh water. So because it's fresh water, there's a suggestion or a possibility that the water potential here is higher than the water potential inside the cell. Okay, so the water potential in the cell is lower. That means that water will frequently move from the environment into the cell. Now, when that water moves into the cell, it goes into a structure called the vacuole. And I think I should use a different color for the vacuole just so it's clear. Okay. It goes into that structure called the vacuole, and when the vacuole is full, or when the vacuole is full of water, it will move to the cell membrane, fuse with it, and then release the water, okay, back into the environment. So basically trying to ensure that the cell doesn't burst or the organism doesn't burst. So a student then decided to count the number of times that the vacuole filled and emptied when the cell was in different solutions, so different water potential. So if the water potential is 0, so that's neither here nor there, the rate of emptying per minute was 31. When it was minus 100, which means a lower water potential, then it was 20, and so it goes on like that. And when it was minus 500, it was 0. Which statement explains the pattern observed as the water potential of the external solution decreased? So as the water potential of the external solution decreased, what can we say about the pattern? All right, the water potential gradient between the cell and the solution increased, causing water to move into the cell more rapidly and the contractile vacuole to empty more frequently. That's not true. Like, that statement already is not true, because we can see from the values we've been given, and I'm just going to erase these so you can see that the water potential was decreasing. Okay, and the rate of emptying was also decreasing. So there's no way that answer is correct. The water potential gradient between the cell and solution increased, causing water to move into the cell less rapidly, and the contractile vacuole to empty less frequently. This is incorrect because the water potential between the cell and the solution might have increased or decreased. Well, it actually decreased, it didn't increase. So that also makes this wrong. I don't know why I thought I saw decrease. Okay, let's look at C. I think C and D might be our good sports. So C says the water potential between the cell and the solution decreased, causing water to move into the cell more rapidly and the contractile vacuole to empty more frequently. I'm tempted to empty more frequently, but no, it didn't empty more frequently. It emptied less frequently as you go down, because you can see here that it started to decrease. So this again is not the answer. The water potential gradient between the cell and solution decreased. So for this question, the key words you're looking for is what are they saying about whether or not it increased or decreased, and what are they saying about the frequency of the emptying. Okay, so here the water potential decreased, causing water to move into the cell less rapidly and the contractile vacuole to empty less frequently. And that's what we observed. And so our answer would be C. Because if you look at it, the rate of emptying decreased as the water potential decreased. So that's it. And I spent only 4 minutes and 40 seconds explaining this. So number 18, just 3 more questions to go. Which row shows the correct number of each component of a single chromatid during anaphase of mitosis? So during anaphase, what do we have? We have 1 centromere. So first of all, I'm just going to draw and this was a question someone asked me in the comments. I hope that they watch this video so they can see. This is a chromosome. Okay. If you split this chromosome in 2, which is what happens during anaphase, by the way, you should study your phases of mitosis. This side moves to the opposite pole, this side moves to the opposite pole, which means you are left with something that looks like that. Okay. So these here, this half here is called a chromatid. And to form a chromosome, you need 2 sister chromatids. That's where that comes from, because you need 2 chromatids to form 1 chromosome. Okay, so you have that, and also you have these ends here that are called the telomeres, and they are on each chromatid, so it doesn't matter. Okay, you have the telomeres, they're on each end of a chromosome, the 4 ends basically. So if you have a single chromatid during anaphase, you will have 1 centromere, because that's the centromere there in the center, 2 polynucleotide strands. This would, I'm guessing these are our polynucleotide strands, but there's 2 of them. You'd also have 2 telomeres, so there are 2 end regions. Okay, so that looks correct to me. But now let's see what the other stuff is. 1 centromere, yes, that again is correct. So remember, it's asking you of a single chromatid during anaphase. It's not asking you for a single chromosome. This whole thing here is a chromosome. A chromatid is a different story entirely, okay? It's half of a chromosome. So centromere, you would have 1. Polynucleotide strands, you won't have 4 because you only have 1/2, so it would be 1 and 2. And you won't have 4 telomeres. So I'm just going to cancel out the one that says 4 telomeres. And this one also says 4 polynucleotide strands. It even says 2 centromeres, and that's not true. So the answer is A. Okay. Number 19, some processes are listed. Cytokinesis, differentiation, DNA replication, mitosis. How many of the listed processes occur during tissue repair by stem cells? This is such an unusual question. I have never seen a question like this. And if you're doing the 2022 exams, you might get this question. So during tissue repair by stem cells, just think of the process of mitosis. What happens during mitosis? So before mitosis starts, you have the G1 phase. Okay, you have the S phase, you have the G2 phase, you have mitosis, and you have cytokinesis. All right. During this G1 and G2 and S phase, DNA replication happens. This is another reason why I say CIE is really interesting for students and helps you develop critical thinking, because the questions are not direct. They expect you to have more background detail. So DNA replication occurs during this phase. So you know that for mitosis to happen, you have DNA replication. For tissue repair to happen, you need mitosis. Okay? So DNA replication happens, mitosis will happen. Because the tissue is repairing, obviously it will, what's it called? Because the tissue has undergone mitosis rather, the cells have undergone mitosis, they would also undergo cytokinesis, which means like they split into two cells and generate like new cell membranes and so on. So cytokinesis will happen. The last bit is that it's telling you by stem cells. So if this was just tissue repair, you might not think of differentiation. But stem cells are able to differentiate themselves into different parts of the body that are required. So differentiation is certainly part of it. So how many of the listed processes would occur during tissue repair by stem cells? The answer is 4. Okay. The last question, and then you are free of me. The photomicrographs show cells in various stages of the cell cycle. Which stage is not shown? This is really easy. And I know my students get angry with me every time I say something is really easy because they say I'm the teacher, so I know everything. That's not true. But if I were you, I would just look at this and label them and say, okay, what phase is this? This looks like prophase or early prophase or whatever, but you can just get an idea there. Prophase. I can see my cells here are like moving in like chromatids sort of, so that must be anaphase, and they're at opposite poles, right? That must be anaphase. This is probably telophase because it looks like they're splitting sort of, and obviously when they're splitting into two like this, that's cytokinesis. I'm not gonna write it. Okay, well, I mean, I might as well. So, I mean, what is missing based on the different phases of mitosis? Metaphase. Metaphase is the phase where the cells are aligned in the center, and there's no diagram where they're aligned in the center. So metaphase is the right answer here. And yeah, that's it. I hope you found the video helpful, and it sort of helped you see how to start thinking about questions like this. Again, practice these past papers. I can't promise to post videos frequently because, like I said, I've covered all of the syllabus. But practice these past papers, practice these questions, and start to dig deep into your understanding so that you can answer them more accurately. For those of you who started scoring yourself, who scored yourselves rather, or did the test before coming to watch the video and seeing how well you did, I hope you did really well. If you didn't do well, don't lose heart. It takes a lot of practice. Just keep practicing, all right? And if you are doing this paper for practice and you find a couple of questions that are unclear, you can send me those exact questions and I will try to do a video on them. But also send me your exam date because I'm so busy and I might not get to it. But if you send me your exam date and I know, then I can try to schedule it in before your exam just to explain how to think through them. All right, everyone, have a good time and goodbye until next time.